Hi, my name is Ryan Urbanowitz, and today I'm going to be presenting our work on Scikit-FIBERS. This work is motivated by a specific biomedical problem, specifically kidney transplant failure. Where we're trying to match donor tissue with recipients, they're going to be least likely of having issues in terms of transplant rejection. Typically, we focus on markers called human leukocyte antigens, or HLAs, which the body uses to help distinguish self from non-self in regulating the immune response. Allocation systems responsible for matching donor tissue with recipients currently typically focus on antigen-level HLA mismatches in determining good matches. However, in contrast with that lower resolution data, there's been recent interest in focusing on higher resolution amino acid mismatches within the HLA complex to better match donors with recipients in kidney transplantation. But there's still a number of questions remaining with respect to amino acid mismatches in the HLA. For instance, what's the relative importance of individual amino acid mismatches at different positions or within particular regions of the HLA? As well as, can we identify a set of positions that together are most predictive of transplant failure, also known as graft failure? Towards an algorithm to help us answer these questions, we drew inspiration from the field of genomics, which has some methodologies laid out for tackling rare variant analysis, specifically dealing with genetic variables that are rarely present across the population as a whole, so we have a limited power to detect the relationship between those risk factors and outcome. One strategy that's commonly used includes binning and burden testing, where the user would specify regions around or within a gene and count the number of rare variants as a bin within that region. And then we can look at relationships between the count of rare variants in a bin and the outcome of interest in an attempt to increase detection power. However, this approach suffers from a couple glaring drawbacks. The first of which being its reliance on domain knowledge and assumptions about where to draw those bin boundaries. Furthermore, it's possible for rare variants within those bin regions to have different directionalities of effect. In other words, some variants could increase risk of disease, but others could actually be protective. So in summing rare variants within a bin, these effects could start to cancel one another out. We first approached this problem through the development of an algorithm called RARE, standing for Relevant Association Rare Variant Bin Evolver, which we presented at GECCO in 2021. This evolutionary binning strategy focused on predicting binary outcomes, where we used a genetic algorithm to evolve combinations of features that together were more predictive of that outcome. We evaluated this proposed methodology with a variety of simulated datasets and demonstrated proof of principle that this approach could be effective. So back to our motivating problem, we want to develop an algorithm that's able to identify amino acid mismatches that help us stratify graft failure risk. And instead of making biological assumptions, we want to leverage a machine learning method that's data-driven. More recently, we developed a new methodology called FIBERS, based on the RARE algorithm. FIBERS is designed to automatically engineer HLA variable bins that can stratify risk groups of graft failure over time. The bins that FIBERS generates are basically equivalent to OR rules, where the outcome, or then part of the rule, is automatically determined by the condition, or if part of the rule. Furthermore, FIBERS evaluates bins based on their ability to separate risk strata using survival curves. The results of this work were recently published in the Journal of Biomedical Informatics, applying FIBERS to a study of amino acid mismatches predicting kidney transplant failure. Overall, this line of investigation is working towards the development of a new HLA-based AI clinical decision support tool to improve patient outcomes in transplantation. Before we talk more about the FIBERS algorithm, let me first introduce you to survival analysis and survival curves, as well as right-censoring. Here we have an illustration of two survival curves being specified for two different groups. In this case, two treatment groups that perhaps were given different drugs. On the y-axis, we have survival probability, which indicates the proportion of subjects in either group that has survived up to a given point in time, illustrated on the x-axis. So in this example, subjects in the red curve are not surviving as long in comparison to subjects in the blue curve. Notice the short vertical lines in these curves, which represent censoring. The more censoring that occurs, the greater the uncertainty surrounding these curves becomes. On the right, we're going to illustrate what I mean by right censoring. On the x-axis, we again have time, and these vertical lines represent the start of the study and the end of the study. On the y-axis, we have individual subjects in this investigation, where the horizontal red lines indicate a subject that died before the end of the study and we observe their death, while subjects in blue survive past the end of the study, and we don't know exactly when they died. We only know that they survived up until the end of the study, or the time at which we stopped being able to observe them. These data points are considered to be right-censored. because there's some uncertainty in how long they survived after the observed point. Now let's look at how the FIBERS algorithm works. FIBERS stands for Feature Inclusion Bin Evolver for Risk Stratification. Starting with a survival dataset, FIBERS first initializes a random population of OR rules. Then the algorithm starts rule optimization, which takes place in a number of evolutionary training iterations. First, we evaluate the current population of rules. Each rule will determine what instances are put in the high or low risk group, and then we can plot these high or low risk groups using survival curves. Next, we apply the log rank test to evaluate the separation between these two curves as a way to quantify difference in survivorship, and this score becomes the fitness of a given rule. Next, we move on to rule discovery using a basic genetic algorithm. This begins with parent selection from the current population, and then the application of crossover and mutation mechanisms to create new offspring rules. Next, we eliminate rules from the population with the lowest fitness, leaving behind an elite set of rules. These are combined with the offspring rules to form the next generation of rules that becomes the new population. And this cycle continues for some user-specified number of training iterations, after which we have a final set of rules that undergo a final evaluation, and FIBERS outputs a summary of the rules and their risk stratifications. How this algorithm works is effectively the same as the paper that we recently published in the Journal of Biomedical Informatics. Let's take a quick look at what the data might look like, as well as a rule discovered by FIBERS. Here we have a small mock data set where rows are instances that represent donor-recipient pairs of transplantations that took place, and columns indicate features in the data set. The column of time is a time to event, either the time to death, if censoring is 0, or the time until a subject stopped being observed if censoring is 1. In this illustration, we have an example FIBERS bin that was discovered that specifies features 1, 2, and 4. Therefore, in the data, if an instance has a mismatch at any of these positions, they'll be assigned to the high-risk group. Otherwise, they'll be assigned to the low-risk group. Once a bin is used to separate out these risk groups of instances, the algorithm will use the log rank test to evaluate the separation between high and low risk. In our first paper on FIBERS, we compared the top FIBERS bin that was discovered to a traditional antigen mismatch model that is typically used in kidney transplantation allocation. And we found that these survival curves were very similar, and that the top amino acid mismatch bin discovered by FIBERS successfully identified risk of graft failure even after adjusting for all covariates and antigen level mismatches. This finding suggests that higher resolution amino acid mismatch data can provide more information about potential graft failure than antigen mismatch data alone. Potentially more interestingly, when we looked at the number of instances in the data that were categorized as low risk versus high risk, we found a much larger proportion of individuals put in the low risk group when using the FIBERS bin, rather than antigen level mismatches. Downstream, this might lead to greater flexibility in kidney transplant allocation. Focusing on the current work presented in this paper, we've first and foremost taken the FIBERS algorithm and implemented it as a scikit-learn compatible software package. And we did this to promote accessibility and comparability as open source software. In setting up this new implementation, users can apply Scikit-FIBERS as a feature learning algorithm using the standard scikit-learn transform method that's been implemented. In this way, users can apply Scikit-FIBERS to identify a population of candidate bins, which each represent newly constructed features, which in turn can be fed into other traditional machine learning modeling algorithms. Alternatively, users can apply Scikit-FIBERS to modeling, using the standard scikit-learn predict function included in Scikit-FIBERS. When this is applied, Scikit-FIBERS will automatically take the best scoring bin it discovered and use it to predict risk group. However, beyond just implementing Scikit-FIBERS, the current study also focuses a lot on simulation. First and foremost, how do we effectively simulate right-censored survival data? Second, we wanted to confirm the efficacy and fidelity of our new Scikit-FIBERS implementation in contrast with the original FIBERS algorithm. The current study represents the first time a controlled simulated evaluation of the FIBERS algorithm has been conducted. And in this simulation study, we take a look at the impact of censoring and mismatch frequency rates in simulated data on the performance of FIBERS, as well as the impact of noise and the overall feature space size. The method we implemented to simulate right-censored survival data is reasonably straightforward. First, we simulate predictive features in the data, where all predictive features that will become a part of the low-risk group are given a value of 0 mismatches, and those that will belong to the high-risk group are given 1 or more mismatches. Next, we randomly simulate non-predictive features in the dataset. The time to event variable is chosen from a Gaussian curve with a mean and standard deviation for both low and high risk groups set independently by the user. where low-risk group individuals are going to tend to get higher time values, and higher risk group will get lower time values, where subjects would be surviving a shorter amount of time. Lastly, we assign the censoring value to each instance, where low-risk group individuals are much more likely to be censored than high-risk ones, since low-risk group individuals survive a longer amount of time. Here's a schematic of the overall simulation study conducted in this paper. We began by looking at dataset combinations with different mismatch frequencies in the data, as well as different censoring frequencies. And as you'll see, we identified a combination of parameters that made a dataset that was pretty easy to solve, which in turn became our positive control. We then shuffled the time to event and censoring columns of that positive control to create a negative control simulated dataset. Once we'd established the basic functionality of the algorithm, we then looked at increasing noise in these simulated datasets and its impact on performance. In the paper, you'll see that we specified two different approaches that we use to try and simulate noise in these datasets. And lastly, we looked at the effect of feature space size on the FIBERS algorithm, simulating datasets with progressively larger number of total features. Here are the basic hyperparameter settings used by Scikit-FIBERS throughout this study. Turning to the results, let's first look at the impact of mismatch frequency range on FIBERS performance. We tested a variety of ranges going from 0 to 0.1 all the way up to 0.8 to 0.9. Here we show a close-up of 4 of these plots in particular. Each plot shows the performance of FIBERS in separating risk groups on the top bin that FIBERS algorithm discovered. With a very low mismatch frequency range, you can see that FIBERS can't separate these curves at all. And as we increase this range, we can see a much stronger separation emerge. And looking at all of the simulations, we can see that at some point, we basically flatline in how strong a separation we can get just by changing this mismatch frequency range. For the rest of these simulations, we chose a mismatch frequency range of 0.4 to 0.5, which gave us a nice separation between survival curves. Next, we take a look at the impact of changing censoring frequency. We observed censoring frequencies from 0 all the way up to 0.8. When censoring was 0, notice how both of the survival curve have no uncertainty in these plots. In other words, no shaded bands around the lines. Since the low-risk group in blue is more likely to be censored, we see greater uncertainty here when the censoring frequency is lower, and when censoring frequency is higher, we see uncertainty growing both in the low-risk and high-risk groups. Moving forward, our simulated datasets adopted a censoring frequency of 0.5. Next, we generated our positive and negative control datasets, again using a mismatch frequency range of 0.4 to 0.5 and a censoring frequency of 0.5. On the left, we have our positive control, where we see a nice separation of our two survival curves. And on the right, we have our negative control, illustrating that the shuffling of our time and censoring variables led to a complete removal of the ability to FIBERS to find a separation between survival curves. Looking at the scores and composition of the top bins discovered by FIBERS in both the positive and negative controls, first we find that in the positive control dataset, we get very high log rank scores, and all of the rules include only features starting with P, indicating that they were simulated as predictive features. Over here in the negative control, we see much lower log rank scores, and most of the features included in bins start with the letter R, indicating that they were randomly generated and non-predictive. Together, this simple experiment illustrates the efficacy of the FIBERS evolutionary algorithm to identify what features to put together in a bin that enable us to separate out risk groups in survival data. Next, we turn to the impact of adding noise to these simulated datasets. Here we'll show the results of just one of our strategies we tested in the paper to simulate noise. In this experiment, we tested noise frequencies from 0 all the way up to 0.5, which was basically equivalent to randomly shuffling the outcomes. We can clearly see a decrease along the way in the top log rank scores identified by the top FIBERS bins. And we can see that better illustrated in these survival curves, going from a noise frequency of 0.1 all the way up to 0.5. Interestingly, if we focus on the simulation with 0.4 noise, which is pretty high, and we look at the top bins identified by FIBERS, we find that while their log rank scores are indeed a bit lower, this algorithm is still very nicely identifying just predictive features in the dataset and not including any random features in the dataset. Lastly, we took a look at the impact of increasing the feature space in these simulated datasets. First, we set up simulations with datasets with a number of features ranging from 50 to 500. And in all of these experiments, we kept all hyperparameters the same, including a fixed max population size of 50 rules. And we found the algorithm performed pretty well until about 300 features, after which FIBERS did not succeed in learning bins that separated the survival curves. In a follow-up analysis, we looked at simulated datasets with even higher numbers of features, ranging from 600 to 1,000, but this time we allowed the maximum rule population size in each experiment to equal the number of features in that given experiment's dataset. And here, Fibers was able to successfully separate the risk curves in all examples. Notice that all of these results have a log rank score above 2,450, which would be somewhere around here. Of course, this work is far from done, and our ongoing work is first focused on expanding the Fibers algorithm flexibility, exploring ways to allow the algorithm to identify the best risk threshold for itself, rather than assuming a risk threshold of 0 mismatches as the ideal cutoff. We're also exploring ways to allow Fibers to adjust for real-world covariate information during the course of algorithm training in an attempt to find even better rules and bins downstream. And also, of course, we're going to be following up with further simulations and real-world applications of these methodologies, specifically within the context of kidney transplantation research. In the meantime, if you're interested in trying out Scikit-FIBERS, you can find it through this QR code, which will take you to this GitHub link at URBS-Lab/Scikit-FIBERS. I'd like to thank all the authors on this paper, including first author Harsh Bandhay, and all of our collaborators, and of course I'd also like to acknowledge our sources of funding.